Hello friends, in this video we are going to discuss about the double titration. In order to understand the double titration, we have taken a solution, and in this solution suppose we have sodium carbonate and 0.01 mole of sodium carbonate is present out there, and 0.01 mole of sodium bicarbonate is also present in it. And inside that one we have added phenolphthalein, and since we have added phenolphthalein, that's why the color of the solution is pink, since the solution is basic in nature. Now we are titrating this solution with the HCl having the concentration of 1 molar, and the thing that we need to calculate here is the volume of the HCl, which is having the concentration of 1 molar, required to change the color of the phenolphthalein from the pink to colorless. And remember that since the solution is basic in nature, why it is basic in nature is because it is a solution of a salt which is made of strong base and a weak acid. That's why the solution is basic in nature and the phenolphthalein is showing pink color. So we need to calculate the volume of HCl having the concentration of 1 molar required to change the color of the phenolphthalein from pink to colorless. So let's start. The reaction that is involved in this particular titration is, first of all, as soon as we'll start adding HCl, the sodium carbonate will react with the HCl, and this one produces sodium carbonate, sodium bicarbonate, and NaCl will going to be produced. Next, whatever the sodium bicarbonate is produced and which is there present in the solution, both of them. Again, I am saying this sodium bicarbonate is the sodium bicarbonate which is produced from this reaction and which is already present in the solution. So both the sodium bicarbonate will come in reaction number 2, and it will react further with the HCl in order to produce NaCl plus CO2 and the water molecule. Now you have to remember one fact. The fact is that when the reaction 1 will get completed, then phenolphthalein will change its color, and when the reaction 2 will get completed, then methyl orange will change its color. The pH when the reaction 1 is getting completed will be such that it is very much suitable to change the pH of the to change the color of the phenolphthalein, and when the reaction 2 is completed, then the pH of the solution is such that in that condition methyl orange will change its color. So phenolphthalein will change its color when the reaction 1 will get completed. So from there we can say that phenolphthalein will change its color when reaction 1 will completed. And in the reaction 1 we can see that 1 mole of sodium carbonate is reacting with 1 mole of HCl, or 2 moles of sodium carbonate will react with the 2 moles of HCl, or 3 moles of sodium carbonate will react with the 3 moles of HCl. That means number of moles of sodium carbonate will be equal to the number of moles of HCl. Number of moles of HCl will be equal to the number of moles of sodium carbonate. And suppose the volume of HCl that has been required to change the color of the phenolphthalein from pink to colorless is V ml. Then the number of moles of HCl required is V by 1000. This is the volume of the HCl that has been required in liters multiplied by its molarity, and that will be equal to the 0.01, which is the number of moles of the sodium carbonate. V by 1000 is the volume of HCl required. It is the volume of HCl required in liters, this full, and V is the volume of the HCl required in milliliters. So from there we can calculate the volume of the HCl that will be required to change the color of the phenolphthalein from pink to colorless will be equal to 1000 multiplied by 0.01, that is 1000 into 1 by 100, and that will be equal to 10 ml. So 10 ml of the HCl will be required, 10 ml of HCl whose concentration of 1 molar will be required to change the color of the phenolphthalein which is present in a solution having 0.1 mole of sodium carbonate and 0.1 mole of sodium bicarbonate. So this is the double titration with respect to the phenolphthalein. Now we'll try to understand the double titration with respect to methyl orange. For methyl orange, we'll consider this problem. Again, we are considering a solution which is having sodium carbonate and sodium bicarbonate. 0.01 mole of sodium carbonate and 0.01 mole of sodium bicarbonate is present. And again, the solution is basic in nature. That's why methyl orange is showing the color which is red. That's why I have drawn such a solution which looks like red in color. Now we are titrating that solution with respect to HCl, which concentration is equal to 1 molar. Now the question is we need to calculate the volume of HCl that will be required, when the methyl orange will change its color from red to yellow. Okay, so again we have to write the reaction. First of all, sodium carbonate will start reacting with the HCl. It will form sodium bicarbonate and NaCl. And second, sodium bicarbonate. Now again I am saying sodium bicarbonate which is coming in the second reaction, it has two sources. One is from reaction 1 and the other one is already present in the solution. So from the second reaction, NaCl, CO2 and water will produce. So the next thing that we need to do here is the number of moles of HCl in reaction 1 and the number of moles of HCl in reaction 2 total will be equal to the number of moles of sodium bicarbonate in reaction 2 and the number of moles of sodium carbonate in reaction 1. This is reaction 1 and this is reaction 2. I have already told that when the reaction 2 will get completed, then only methyl orange will change its color, because then only the pH will be such that which will make sure that the methyl orange is changing its color. So both the reaction has to be completed in order to make sure that methyl orange is changing its color. So we need to consider both the HCl of reaction 1 and the HCl of reaction 2. So from equation 1 and 2, or from the reaction 1 and 2, we can say that number of moles of HCl will be equal to number of moles of HCl will be equal to number of moles of sodium carbonate plus number of moles of sodium carbonate in reaction 1 plus number of moles of sodium bicarbonate plus number of moles of sodium bicarbonate in reaction 2. And I am repeating again, the sodium bicarbonate which is coming in the reaction 2, it actually has two sources, one from the reaction 1 and the one which is already present in the solution. So suppose V ml of HCl is required completely in order to make sure that methyl orange is changing its color from red to yellow. That's why I am writing V ml. I am converting into the liters by dividing it by 1000 and the molarity of the solution is 1. So this is the number of moles of HCl total required. Now what is the number of moles of sodium carbonate in reaction 1? Then that is 0.01. And again, number of moles of sodium bicarbonate in reaction 2, it has two sources, that is number of moles of sodium bicarbonate coming from reaction 1, and plus number of moles of sodium bicarbonate, already present in the solution, So V by 1000 into 1. This is the number of moles of HCl, 0.01. It is the number of moles of sodium carbonate. Now the number of moles of sodium bicarbonate which is coming from the reaction 1. See here, in the reaction 1, whatever the sodium bicarbonate is coming, it is coming from sodium carbonate. So number of moles of sodium bicarbonate which is coming from the reaction 1 will be equal to the number of moles of sodium carbonate itself. So that is also 0.01, and the number of moles of sodium bicarbonate which is already present in the solution which is given in the question, which is 0.01 as well. So this will become 0.03, and from there we can calculate the volume. The volume will become 1000 multiplied by 3 by 100, that will give 30 ml. So 30 ml of HCl having a concentration of 1 molar will be required to change the color of the methyl orange from red to yellow, which is present in a solution which is having 0.01 mole of sodium carbonate and 0.01 mole of sodium bicarbonate. So this is the one very good example of double titration. So from this example we can understand the double titration, I hope. Thank you.